So now we're going to go to one of the two finalists for our top research award for clinical research. And it goes to Dr. Brian Clemency on efficacy of inhaled CSTI for outpatient treatment of adolescents and adults with symptomatic COVID-19, a randomized clinical trial. Congratulations, Brian, and go ahead.
Thank you. Our study was published in JAMA Internal Medicine in November of last year. Our disclosures, this was an industry-sponsored study, and I was lucky enough to be in the family medicine K-12 program while the study was being conducted. I need to acknowledge my co-investigator from the University of Buffalo. Ranaj Varghese is a junior faculty member, an absolute dynamo, who was the second author on this paper, as well as the phenomenal mentorship team from the K-12 who are unbelievably supportive of me, both as a researcher and as someone trying to expand my academic career. Aceclestinide is an inhaled glucocorticoid. It's used to treat asthma and allergies. It is used much more in Europe and just is not commonly prescribed in the US, although it is FDA approved for those same indications. There were some early case studies in 2020 that described the use of this steroid in patients with COVID-19. And there was also some evidence that chronic steroid use was protective against severe COVID-19 diseases. Some patients with asthma or COPD who we thought might be at higher risk if they were on inhaled steroids didn't seem to be higher risk for disease progression after all. And there was some Petri dish evidence that it inhibited coronavirus replication. And so this was one of the drugs that already existed that was identified as being of potential interest for use in COVID-19. This was a multi-center randomized controlled trial with 10 sites throughout the US. The medication was aceclestinide via MDI, 320 micrograms, 2 puffs twice a day for a total daily dose of 640 micrograms, which is the top area of dosing for this medication for other indications. The placebo arm was an identical saline mist MDI, and patients were instructed to take the medication for 30 days, even if their symptoms got better. So this audience is made up of researchers and problem solvers, so I thought I would touch base on some of the challenges that we faced and how we overcame them to bring the study to Buffalo. So our first challenge amid COVID, remember it's June 2020, was where are you gonna go? Every time we talked about the study to someone, we got the same two responses. This is a great project, you should totally do it, but not at my site. Right? People were not tripping over each other to bring new COVID patients into different areas. Our second major challenge was the unbelievably short enrollment window. So we needed to get patients having a nasal swab, having their results, recruited for the study, consented for the study, and have their first dose of medication within 72 hours. This was at a time when many people were not getting their results back until 96 hours. And so, in fact, more than half of our patients were enrolled in the final 12 hours or so of their potential enrollment window. And so when you have these challenges, you need to try to figure out how to overcome them and how to pivot to your strengths. So the Department of Emergency Medicine has over 2 decades of telemed experience, and we're also the medical directors for AMR and many other EMS agencies. We also had some help from the CRO office and an unbelievable department chairman in Dr. McCormick. So here's how we made this work. We knew we couldn't bring patients to us in such a short timeline, so we went to them. So AMR leased us EMTs who were trained as members of the study team. They completed all the city trainings and GRP trainings, study-specific trainings, and all of them were trained to nasal swab and used me as their training mannequin, unfortunately. They would don PPE and go to the home of the patient. They would set up a telemed visit, get vital signs, patient demographics, height and weight, witness a consent, and teach the subject how to do some of the parts of the study. We'd have a study nurse there virtually, that they'd be responsible for data collection and many of the other study components, and there'd be a study physician also there remotely for the process. Once the subject was in the study, the protocol called on them to twice a day check their pulse ox with a pulse ox we gave them, take 2 puffs of their medication, and log their pulse ox their medication use, and their symptoms into a mobile app. We also called them 8 times throughout the study month on an every other day schedule for the first week or so, and then more spread out after that to check on them and see how they were doing. At the end of 30 days, we sent the same EMS crew back to their home. We collected their study samples. We gave them another nasal swab, and one of our study nurses did another telemed visit with the patient. Think back to 2020. Remember, in January, we had the first case in the US. In March, we saw the first case in Erie County, and this red line shows our cases in Western New York. We, of course, enrolled during the trough. We stopped enrollment of this national study in the beginning of November, just as numbers took off and the first monoclonal EUAs came out. The key outcome measures we were looking for was time to alleviation of COVID-19 related symptoms. And this was designed as defined as logging into your app, 3 consecutive BID registrations. So, every 12 hours times 3, and saying you were symptom free. Once you did that, that was your time to alleviation of symptoms. And we asked about a handful of different symptoms, such as fever, chills, difficulty breathing, sore throat, changes in taste and smell, and cough. The secondary analysis looked at percentage of patients with subsequent ED visit or hospital admission due to COVID-19, or percentage of patients with hospital admission or death. Nationally, the sample included 400 subjects total, roughly balanced between the treatment and control arm. If you take a look at patient demographics, they were pretty similar between the two groups, except that there were more type 2 diabetics in the treatment arm, and there were also more patients with asthma in the treatment arm. But because being on an inhaled steroid already was a contraindication to entering the study, the only patients with asthma in our study were those who were not on inhaled steroids already, which was probably a proxy for patients with mild disease. It probably excluded patients with more severe COPD or more severe asthma. There were no subjects that died in the study. And in the end, our sample consisted nationally of many young and otherwise healthy subjects. Unlike some of the monoclonal antibody studies where they had specific criteria that made you high risk for enrollment, we took all comers who had COVID, could be enrolled within 72 hours, and did not have any of the exclusion criteria. So for the primary outcome, there was no difference in median time to resolution of all symptoms between the two groups. It was about 19 days in both groups. And you see a little bit of separation as you come out past 19 days, but that was not statistically significant. Feeling better and faster is nice, but I would argue that during a pandemic, staying alive and staying out of the hospital is probably even more important to both the health of the patient and the health of our healthcare system in general. And so the number of patients in this group with this outcome was actually below what we expected. Our patients did much better than people who designed the study thought they might do when we thought about this in April of 2020. Again, unlike the monoclonal antibody studies, this study enrolled all patients, including many, many healthy patients, and that probably drove down our rates of bad outcomes. When we looked at our data in a post hoc analysis that's not included in this paper, but that we're looking to publish in the future, when we take a look at all our patients and we place over our enrolled patients a second filter of would you meet the original monoclonal antibody criteria, that cuts our sample size in more than half, but all but one of our patients with a bad outcome remain in that group. So we substantially cut the denominators of both groups while leaving the numerators fairly similar. And so you see a larger relative effect size between those two groups. Similar findings have been found for budesonide, another inhaled steroid. And so it seems like inhaled steroids may be a way to not necessarily make you feel better faster, but stay out of the hospital longer. And finally, my 10-year-old tells me that I'm not cool unless I have a meme. I tell her that the equivalent of a meme for a clinical researcher is a visual abstract. She is not impressed by this at all, but I will show you mine anyway, and I'll be happy to take any questions.
Excellent. So yes, we're open for questions here.
Go ahead, Steve.
Great. Brian, great study. So important. I have 2 or 3 things that come up thinking about this. We currently have an active study on Paxlovid in children, and and as we were putting it together, in adults, there's at least 4 different treatments for outpatient COVID in adults where the drug costs per hospitalization were below Medicare estimated hospitalization costs. One is, as you're bringing up, inhaled corticosteroids, but I'm wondering if you controlled at all for Paxlovid use, fluvoxamine, colchicine, because all of those were confounders potentially in outpatient reduction in hospitalizations for adults with COVID. And then another quick question is you had such a broad range of ages, 13 to 87. In kids, sometimes there's mixed results of inhaled corticosteroids for respiratory viral diseases, including COVID. I know you had relatively small number of kids, but do you think you had, by having such a broad range of ages, maybe you controlled for some underlying disease, but maybe not all? And then the other thing is you brought up budesonide, and the TACTIC trial really showed that inhaled budesonide really reduced disease progression in adults with COVID, but that was more in hospitalized. So do you think, again, it's it might be not just patient selection, but stage of disease or some of these other factors that might have affected your results?
So absolutely. So there's 3 questions, and I'll try to remember them all as I go. So first, a number of those other agents were either not in play in the early part of the pandemic prior to the monoclonal antibodies, or were not frequently seen in our patients. So the vast majority of our concomitant medications were bread and butter things like antihypertensives and Tylenol and antipyretics and those kinds of things. There were almost no patients on any kind of antivirals or anything else like that, as many of them were relative exclusions. As for, is this the right medication, or what do we think about relative costs of these things? Budesonide has shown in a number of different studies, as have some of these, has Ciclestinide and other studies, to be helpful in people who are at higher risk. If I enroll 300 18-year-olds with COVID, the chance that any of them are going to have adverse events, if you know nothing else about the population, is pretty low, right? We know, well, certainly individual children and individual young adults have had terrible catastrophic outcomes from COVID. On a macro level, children do very well if they get COVID. Young, healthy adults do very well if they have COVID. And I think the highest potential benefit for this is patients who either have pre-existing conditions that make them more likely to have an adverse outcome, or have a current clinical presentation that makes you really worried about them, right? An 18-year-old who has no comorbidities that you know of, but looks terrible, is somebody who's at high risk. And so I think what is missing from some of the discussion about monoclonal antibodies and other interventions is not just risk factors, but clinical gestalt. Because we're going to find some patients, like a patient in our study, who we did not know he had any risk factors, and he didn't know he had risk factors, but he retrospectively realized that he had kidney failure. And that led to a hospitalization relative to COVID. And so I think it's both clinical gestalt and risk factors that we need to think about as we're prescribing medication.
Well, that's the point I was getting at in terms of trying to do sub-analyses at some. I am sure that for certain populations, your treatment would probably be effective. So the question is, how do you define who those populations are? Yeah.
So what we're looking to do in our secondary analysis is, I don't want to make up a subpopulation, right? I think that's fraught with lots of potential downsides. And so what we're looking to do in our next paper is take a look at the published EUA for monoclonal antibodies, take that exact scheme and apply that as another inclusion-exclusion criteria to the patients we enrolled. And when we do that, we get rid of a lot of healthy patients, and we get rid of very, very few bad outcomes, because almost all of the bad outcomes that we saw were in patients with known risk factors based on the initial monoclonal antibody EUA.
Excellent, thank you. All right.